Good morning, everyone. I'm John Leiper, and I'm going to be talking about really focusing on the gastric emptying and intestinal absorption, which is the area of which I got some experience in. First of all, though, I'd like to thank the European Hydration Institute and specifically Ron for the kind invitation to attend this very interesting, I'm sure, meeting, and also for the opportunity to present this short talk. For most people, the alimentary tract is where water comes in. We lose water daily, and we've got to replace that water loss. And for the majority of us, we ingest more fluid than we actually need, and we excrete the extra over it. The majority of intake is really dependent on habit for most people. If you change their environment, change that habit, they sometimes become dehydrated. But there are various things that will help encourage people to drink. One is obviously taste of the drink. So the alimentary tract itself, it's a conduit for the intake of fluids. There are receptors in the upper part of the tract that encourage drinking, taste, dry mouth, as my mouth is very dry at the moment, and throat, sticky saliva, those sort of issues encourage people to drink. There are receptors in the tract that meter the water fluid intake from individuals. It's not quite decided how that works fully, but in doing that, the effect of satiety is triggered. Gastric fullness also is a potent satiatal response. Now, further along in the gut, the actual conservation of water and electrolytes, it occurs mainly in the small intestine, with the jejunum and ileum being the main regions of conservation, with the colon being mainly a functional reserve for most people. Ingesting fluids, they immediately go, quite quickly move into the stomach, which is effectively a reservoir. And at that point there's no real net water or solute absorption in the stomach. The material has got to move into the small intestine in order to be absorbed. Gastric emptying is a highly regulated mechanism. It releases fluid and nutrients into the duodenum in a controlled manner. In the small intestine, where the absorption occurs, there are specific transporters that absorb the nutrients and electrolytes from the intestinal lumen. Water uptake is a passive process and is dependent on the establishment of suitable osmotic gradients. These are some of the factors that are known to affect gastric emptying or not to affect it. Most of this information has been derived from intubation studies, where tube is passed into the stomach, and the rates of emptying have been measured. Volume is an important factor. Increasing the intragastric volume increases the rate of emptying up to a maximum capacity. And this capacity appears to be different for each individual. Increasing energy density of the beverages slows gastric emptying rate quite considerably. And we can note differences between a 2% and a 6% carbohydrate solution quite easily. Increasing osmolality of the beverages also decreases emptying rates, but the effect of osmolality is much less than that for energy density. Temperature has little effect because for most beverages, the collaboration time in the stomach is relatively short, in a matter of a few minutes. Again, pH of beverage has little effect on emptying rates, again because there's limited buffering capacity in most drinks. Exercise can affect gastric emptying. Low intensity exercise does increase gastric emptying very slightly compared with sitting sedentary emptying. Steady state exercise above 70% of VO2 max slows gastric emptying, but so does short and small amounts of high intensity intermittent exercise, which specifically and measurably can slow emptying. As I say, most of these factors here have been derived using intubation techniques, but there are many other different techniques that have been used. Validation has been quite difficult for quite a few of them. I wouldn't say that most of them have been properly validated. But having said that, most methods can be used to make comparisons between beverages using the same subjects and the same methodology. The between subject variability in gastric emptying measurement is quite high. It's about 30% in most studies that have looked at this here. But if you look at the within subject variability, it's much less, at around about 10%. Moving on to the small intestine where the absorption occurs, it's the structure of the small intestine is such that it is designed quite well for absorption. It's a large absorptive structure with the villi increasing the surface volume. There's a single layer of columnar cells, the enterocytes, that line the surface of the villi, so limits the delay in transferring material that has been absorbed. The microvilli increase the absorptive surface. They are on the apical surface of the villi. But more importantly, probably, is the fact that the digestive enzymes, the main digestive enzymes, are bound to the microvilli. They break down the complex nutrients, making smaller molecules that are more easily absorbed. And structurally quite close to these digestive enzymes are nutrient transport carriers of different specificities that translocate the nutrients across the enterocyte membrane. There is also modifiable intercellular tight junctions between the enterocytes, which allows water and some solutes to pass through. There's a rich blood supply with an extensive network of capillaries just below the epithelial cells of the villus, which allows movement of the absorbed material rapidly into the circulation. The main transporters are carrier mediated. They are, as I say, microvilli bound, and they translocate the solute from the lumen to the enterocyte cytosol. These carriers are specific to individual molecules or to molecules of a similar structure. There are active transporters that use the electrochemical potential of cations, and in humans it's mainly sodium that's cation that's used, and I've given the example here as the sodium-dependent glucose transporter, SGLT1. There is also diffusional facilitated transporters such as GLUT5, which is the main carrier for fructose. These are energy and sodium independent, although, well, I should say it was cation independent, but in humans it's sodium independent. But they are concentration dependent, and although the uptake using these facilitated transports is faster than simple diffusion, it's much slower than active transport mechanisms. There is also electrically neutral ion exchange mechanisms and ion cotrans systems cited in the enterocyte membrane, which allows for electrolyte absorption. Important thing to remember is that water absorption is a passive, or mainly a passive consequence of the establishment of suitable osmotic gradients that drive the uptake of water from the lumen across the enterocyte. And the osmotic gradients are produced by the active uptake of nutrients and sodium that produce the greater osmotic gradients in man. Facilitated uptake is less effective than active co-transport because there's more osmotically active molecules that are transferred and they're transferred faster. But if all the active uptake carriers are activated, then facilitated uptake may be given additional effect. Aquaporins are the integral membrane pore proteins that act as highly efficient water channels. And in man, the aquaporin AQP10 is concentrated in the apical cells of the villi, and they effectively help the water uptake along the osmotic gradients. And these aquaporins in some isoforms may also allow the uptake of some solutes. Water and solute can also pass between the enterocytes, between the tight junctions, and this is because the perijunctional actomycin ring in these junctions is activated. It can be activated to increase or decrease the permeability. Such things as glucose and actively transported amino acids activate this response and increase the permeability, allowing water flow by osmosis through the tight junctions, and along with that, some solute is thought to be carried through also. Looking at factors that affect gastric intestinal absorption, most of these factors have been identified using steady-state perfusion techniques, where a tube is passed into a section of the small intestine. Steady-state conditions are established along that segment of gut, and they look at the effect of altering the beverage or the perfused fluid materials in order to look at effects on absorption. Gastric emptying rate is obviously an important one. Increasing the rates of emptying increases intestinal absorption. Osmolality, moderate hypotonicity in the lumen appears to enhance water uptake, as might be expected, but strangely enough, it looks as if the difference in hypotonicity levels is very small, and osmolality between 200 and 260 milliosmoles per kilogram appear to be optimal in humans. Hypertonicity, on the other hand, that is greater than about 300 milliosmoles per kilogram, decreases uptake there, compared with isotonic solutions. Carbohydrate content is important, with the glucose sodium cotrans promoting water absorption, with fructose being less effective in that there, and the amino acids that are actively transported are also effective in promoting water absorption. Some studies have shown that a large amount of maltodextrin in the beverage may assist water uptake, because osmolality is much lower per unit amount of carbohydrate using maltodextrins. Sodium concentration, one would think, would be an important factor in active transport that's required, but it's not clear if sodium is really required in the drinks, because the efflux of sodium into the upper part of the small intestine is usually so rapid that, at least in perfusion studies, it's not been shown that sodium in the perfusion material is required. pH, it's unlikely that pH has any effect because the intestinal buffering capacity, it's unlikely that any beverage or perfusion solute would be great enough to alter that buffering capacity. And again, temperature, it's unlikely that drink temperature will affect luminal uptake, because it won't affect luminal temperature. Exercise, you can appreciate using tubes that are inserted into people. It's quite difficult to do exercise in there, but some studies have shown that steady-state exercise at below 70% of VO2 max has little effect on intestinal absorption. But obviously, higher levels and intermittent exercise would affect gastric emptying, which in turn would slow intestinal absorption. As I say, the perfusion technique, steady-state perfusion, is a method that's been used to identify these factors. And in these sort of methods, the between-subject variability is again quite high, at about 22%, with within-subject variability coefficient being around about 12% or less. In order to try and get a more naturally, a method that appears more natural to us, isotopic tracers for water have been used to try and look at the integrated response of gastric emptying and intestinal absorption by labeling drinks which are then ingested. Both deuterium oxide and tritium oxide appear to be appropriate tracers. This has been shown from about the 1950s. They are odourless, tasteless, relatively safe, and more importantly, they are measurable. The thing to remember is that isotopic techniques don't determine the net rates of water absorption. Rather, the comparisons give you the relative rates of absorption of drinks between solutions. And I'm suggesting that the unidirectional flux is the best measure to use in this type of experiments. The accumulation rate of water tracers in the circulation does appear to follow the integrated effects of both gastric emptying and intestinal absorption of these labelled drinks. The technique has been shown to have a between-subject variation of around about 40%, which again is very high. The inter-subject variance hasn't been really looked at as yet. To show the effect of the integration of both gastric emptying and intestinal absorption, I put up this slide here of a study we did some years ago here, looking at 3 solutions with different, the gastric emptying rate was measured using intubation method. I've used T½ time here, the time taken to empty half of the ingested solution. Water absorption was measured using perfusion study, and deuterium accumulation from labelled drink, and looking at the accumulation in the circulation to the maximum concentration. If we look at the 3 solutions, solution A emptied much faster than did solution B or C, which had emptied at approximately the same rate. Water absorption was higher in the solution A compared with solution B, which in turn was higher than solution C. So this gets rid of the gastric emptying effect. That's just the absorption in the small intestine. Looking at the accumulation rate to maximum in the circulation, we can see that solution A, the accumulation rate was faster than it was for the other 2 solutions. It had a faster rate of gastric emptying, faster rate of water absorption, and showed a faster rate of deuterium accumulation. Solution B showed a faster rate of accumulation compared with C, and although the gastric emptying rates were similar, there was a difference of almost twofold in the water absorption, which suggests that the pattern of deuterium accumulation does indeed follow this integration of both gastric emptying and water absorption. And finally, I'd like to just finish with this slide here, which is a very complicated slide, but I'm only going to make 2 points here. Quite a few of the studies that have looked at typical absorption between solutions have used the area under the curve here over 2 hours to 3 hours there. The slope of accumulation in the circulation is the method I suggest is best used here. Ingestion of the solution, rapid rise of the tracer into the circulation as influx from the lumen into the circulation is high. It then levels off, and then there's a drop in the concentration in that where the influx from the lumen is less than the efflux out of the circulation into the total body water pool. And eventually it comes down to a steady state where the tracer has effectively equilibrated with the total body water pool, and we can then measure the water pool there. But this response here, which is the fast response here, I think is the better method of measuring or comparing drinks, because the area under the curve takes into account this huge area here where influx is less than efflux. And I'd like to stop there if that's possible. Thank you.
Thank you for making the chairman's job easy by sticking to time. It's never good to have to tell speakers to stop, but you did very well as far as time. The 2 discussants for this paper are Susan Sheriffs and myself. Do you want to start, Susan?
Can I ask you a little bit about energy density, osmolality, and the isotonicity? Because you mentioned both of those as all of those being influencing gastric emptying and intestinal absorption. But I think I'm correct from your summary that most of our knowledge on that comes from carbohydrate.
Yes, indeed.
So how well can we take that information and say it will be similar or different if we're talking about proteins or fats or indeed solid foods that are contributing ultimately to our water intake, let's say?
I take your point. I mean, I must admit I focused on beverages as such, but certainly there are at least 3 studies I can think of that looked at amino acids or at least dipeptides, and they seem to have the same effect, that it's the energy density that seems to be more important. If you make the same osmolality using electrolytes, then you don't get the same effects. It's slowing seems to be a measure of the energy density of the drink. I mean, again, I couldn't say anything about fats. I don't think that's really been looked at as such. I'm not aware of anything on fats. But certainly proteins and carbohydrates seem to have an energy density effect rather than osmolality effect. How it's measured, how it's done by the body, because most of this material obviously has not been absorbed. So how does it work? I don't think we're at all clear about how that. I mean, there's been various suggestions that so much carrier gets activated in a certain length of gut. Well, maybe that puts a feedback effect, but you know, it's speculation at the moment.
And just following on from that, on the hypertonicity issue then, so the 200 to 260 stimulating maximum water uptake, that, I'm assuming from your answer there then, that a significant amount of that tonicity contribution is from either the carbohydrate or protein rather than electrolytes. Will it be different if it's, let's say, just electrolytes that are making up that 200, 260?
Yeah, well, I mean, absolutely. I mean, if Nancy Herrera did a couple of studies in which she looked at using electrolyte solutions only, and the absorption rate from electrolyte solutions is quite slow, even though they're hypotonic. Although most of the things that she did was looking at isotonic solutions, but some of them were indeed hypotonic. What she had in most of her studies was at least some carbohydrate, and then altered the osmolality using electrolytes. And certainly electrolytes had very little effect on absorption there.
Mm-hmm. Yeah. Just a general question on the title. Your title was about the role of this in hydration status, basically. Where do you see the gastrointestinal tract's importance relative to, say, other organs, renal system, and so on?
I think it's. I mean, I would have to say I see the alimentary tract as being very, very important. For a start, it's where you stick it all in. But there's a whole variety of things. I think gastric emptying and intestinal absorption. I think it's been shown in plenty of clinical studies that that's important. Having a stomach is really helpful, as is having a reasonable length of gut. Small intestine is very helpful. So I think it's part of the whole integrated system of fluid control. For the majority of people, as far as I can see, they drink more than they actually need, and there is plenty of time. It's episodic drinking for most of us, anyway, and the gut holds the drink. It allows slower release than if we were putting it intravenously, and I think it's beneficial.
Okay, shall I carry on, or do you want to come?
I could maybe dive in and ask a couple of ways. You highlighted a very large variability for all of the measurement techniques you described: gastric emptying, intestinal absorption, and the deuterium tracer. And the variability was greater between subjects than it was within subjects, but it was still quite high within subjects. So we've got two separate things taking place. What do you think the difference between individuals is that accounts for that variability?
Well, I think there is genuine variability in gastric emptying rates in individuals, and it may come down to their methods of eating and drinking. If I can cite John Robertson, who was a postgrad in our department many years ago. He was involved, he was quite a decent runner. He wanted to get into the London Marathon, He was trying to make a time to get into the London Marathon, and we, being physiologists and very helpful, we encouraged him to drink heavily when he was running, get as much fluid into him as he could when he ran. And his times got worse and worse and worse. So we then, we were doing a study in the department. Most of the subjects we used were normally staff within the department and students. He volunteered for the gastric emptying. And whereas majority of people, when they ingest something, the gastric emptying rate follows a negative exponential curve, he did nothing. He emptied nothing for almost an hour, and then everything just came shooting through out of the stomach. If you looked at his eating habits, he ate every hour. He went and he snacked the whole time throughout the day. He never actually sat down and had a meal that I ever saw. He just snacked. And he had the most strangest form of gastric emptying pattern that I've ever seen. So I think people are different, and we're assuming that this happens when you're a baby. The amount of fluids, the amount of foodstuffs that go in, it activates the carriers, and this mechanism is a developed mechanism in the early stages of life, because it's difficult to see how energy density can affect gastric emptying when that energy hasn't all been absorbed. So I'm assuming it's something like carriers that are activated, the number of carriers that are activated, the hormone responses that are triggered from that they all are integrated into developing the rate at which each individual empties. But I mean, I think for a between subject variability of around about 12%, I think that's pretty good in it. It's not quite as good as using a glass bottle and doing the experiment in a glass bottle, but I think 12% is a pretty reasonable level of variability from day to day. And it may be that even that variability is due to what the person ate the day before or the day before that, affecting the response.
So to what extent do you think it's trainable? If you have somebody who's a slow gastric emptier, who's a slow intestinal absorber, can they do anything to speed up that hydration process? The two experiments that I've seen where people have tried to train the gut, the stomach, showed no difference. We did a study in which people drank a sports drink each day. They drank large volume of it each day. The gastric emptying rate had been measured before they started this. They then drank a litre of sports drink throughout the day, taking most of it in the evening, and there was no change in gastric emptying rate over 4 weeks of this sort of training. What did happen was that they especially the women in the study, they found it less difficult to drink. They could drink it faster, and they didn't get the same huge response to gastric fullness. And Asker Jugendrup's group also did a study using water, taking large volumes of water over a period, and there was no training effect seen there either. So it seems to occur, if it does occur, it occurs as a baby rather than as an adult. But we know with animal models you can induce intestinal transporters in adult life. If you take sheep, for example, that have been weaned and feed them lactose, you will induce the lactase enzyme and you will induce the transporter. So the gut can adapt even later in life.
Yes, and certainly, I mean, studies in humans have shown that if you increase the carbohydrate intake of individuals, they will absorb more quickly that carbohydrate, specifically, and again with fats and with proteins. And it's very specific for whatever you've used to increase the nutrient.
You mentioned metering of water intake with oropharyngeal receptors, and you alluded briefly there to you said you didn't think it was likely you could monitor the carbohydrate, but we know there are carbohydrate receptors in the mouth. Yeah. And a number of studies in the last 10 years or so have looked at putting a carbohydrate drink in the mouth, swirling it around for a few seconds, spitting it out, and it definitely sends signals to the brain, because the MRI studies show regions of the brain lighting up, and exercise performance is enhanced.
Yeah.
So do you think when you normally drink, it passes through the mouth too quickly?
I would say yes, because in most of the studies they spend some time rinsing their mouth with it. But it certainly is a fair point. I mean, it may be that further up in the mouth, in the pharyngeal area, perhaps.
People tell us to swallow those drinks quickly so we don't erode our teeth.
Yeah.
And then my mother told me I should chew my food 47 times or something before
Always
swallowing.
believe your mother.
Keep it in the mouth. But we seem to be giving people conflicting advice.
Yes.
On the one hand, we're saying drink quickly so you get these things away from your teeth and help look after your teeth. On the other hand, we're saying drink slowly so there's time for the physiological effects to be apparent and potentially reduce intake.
I mean, cleaning teeth doesn't take very long, does it? And it's pretty cheap, so I would say take it slowly and then clean your teeth afterwards.
Yeah. Not always very practical.
Dentists actually say clean your teeth beforehand.
Yeah.
Yes,
To
well,
get the bugs up.
Yeah.
So you need to be a bit careful.
I had a professor of surgery who told me that he always washed his hands before he went to the toilet.
But then the people who are interested in looking at nitrate as a mediator of some of the reactions tell you not to clean your teeth because you kill the bugs, and the bugs convert the nitrate to nitrite, which gives you some of the beneficial effects. So whatever advice we give, somebody comes along and says that's the wrong advice. A couple more quick points from me before we move on. You said you didn't think temperature was very important from a gastric emptying point of view, but there used to be lots of reports in the literature that if people drank large volumes of very cold drinks, they got a gastric dumping syndrome, which I've never seen or experienced, but you still see it in some of the textbooks.
Yeah, I mean, I think it's a fallacy. I don't know how it came about. I mean, we looked at these ice slurries and people taking, where we had a thermometer in the stomach, people taking fairly large amounts of these ice slurries, about 600 mils. Within 3 minutes, the temperature of the stomach was above 30 degrees centigrade. You know, I mean, there was an immediate drop, but it came back up very, very quickly. And when we looked at even things like blood flow in the arm, no effect whatsoever, taking fairly large amounts of these slurries, which have got, you know, reasonable thermal capacity in them.
But where temperature maybe does become important is where you need to drink a lot. If you're physically active in hot environments and you need to drink fairly large volumes, it's difficult to drink large volumes of cold drinks.
I would agree, but I don't think it's a temperature effect as such. I mean, in that, I think if you looked at the temperature in the stomach minutes afterwards, then it would be pretty near normal core temperature. But it may be that in taking the cold drinks immediately, you get that response which gives you the splitting headache and responses like that.
Not just the splitting headache. When we try to get athletes to drink 2 or 3 litres of drinks at 4 degrees centigrade straight out of a machine that dispenses them at 4 degrees, they couldn't do it. When we let the drinks warm up to 10 or 12 or 15 degrees, they could happily drink the same volume. Yeah. So I think too much cold can be counterproductive if you want to drink large volumes.
Well, certainly one of the studies we did in Loughborough, where we were looking at gastric emptying, and these individuals drank 500 mils and then they had to drink 300 mils every 15 minutes. I mean, that had no effect on gastric emptying whatsoever, and they took the drink straight out of the cold in the ice baths that were used there. But yeah, perhaps if you're talking about litres, perhaps that is a
last comment from me. Given that most people take drinks together with food, I might suggest all of this stuff is completely irrelevant, because as soon as you mix the drink with the food, the composition changes and therefore the gastric emptying becomes completely different, the intestinal absorption becomes completely different. And in most cases, you probably, rather than getting a hydration in the immediate aftermath, you get a dehydration, because you get a net secretion into the gut. And that's something that people don't seem to be aware of, the fact that if you take something that's strongly hypertonic, in the short term, you'll secrete water into the gut.
Yeah, I mean, I would totally agree. I suppose I've come from an area of sports drinks where people don't eat during exercise, that they drink. But yes, I mean, There's no doubt about it that the presence of food, particulate matter, slows gastric emptying and therefore will slow intestinal absorption. And for most of us, that's the way we take our fluids. We take it along with food, whether it's tea and a biscuit or whether it's a meal with a glass of water or a glass of wine.
How long do you think that effect lasts? Because most of the stuff in the literature, whether gastric emptying or intestinal perfusion, has been done after an overnight fast.
Yes.
Would you get the same results if you did it, say, 3 hours post-absorptive, when some of the residue of that food certainly still going to be in the GI tract?
I mean, I think the answer to that would, my feeling would be yes, and it would depend how much was in the stomach, how much was in the intestinal tract. But certainly, I mean, it would change the osmolality, it would change the viscosity, it would change so many different things that would bound to affect intestinal absorption. Yeah.
Susan, do you have any more questions before we open up the discussion?
Just a quick question about the colon, because you had a mention up there that it's functional reserve, possibly its main function. And I think from your manuscript, you had numbers around the lines of maybe something like 600 mils reaches there a day, 400
something like that,
absorbed.
yes, yeah.
But it could have a capacity of 2 to 3 litres?
Yes, yes.
Do we know a lot about it?
Not an awful lot.
Is that based on?
I mean, it's that most of that information is based on studies that were carried out in the 50s and 60s. But there have been a couple of studies that Ralston and that group, Mike Farthing's group, did, where they did they washed out the whole of the intestine and then looked at absorption rates in different segments. And, you know, the colon obviously can absorb quite rapidly fluids. But the 400 to 600 is information that was gleaned way back in the, I think, early 60s. Most of that was there. So, you know, no one's looked at it recently. And a field of experimental endeavour, I would think, in that there. Yeah.
I seem to recall studies from the American military looking at putting rehydration in via the rectum.
Via rectum,
when
yes.
Clean water wasn't available and perhaps the gut wasn't fully functional.
There's no doubt about it. The colon can absorb. I mean, the cholera work that was done by way back in the 1800s, where they threw up a couple of gallons of water up and then put in a cork or a leather bung into the anus. Yeah, I mean. So yes, I mean the colon is perfectly capable of absorbing quite large volumes, but what volume actually goes in normally into the colon per day is perhaps a little bit of conjecture. Yeah.
Okay, thank you.
Thank you, Susan. It's open now for discussion. If you want to stick your hand up and dive in.
John, all other things being equal in terms of temperature, pH, solute load, what about the timing of ingestion during physical activity? And I'm thinking in terms of many small gulps of fluid versus periodic larger boluses if you want to deliver something into the bloodstream.
Well, I mean, certainly the fuller the stomach, the faster the emptying rate will be. And whether there's a large volume in there which you keep topping up would perhaps be the best method of doing it, rather than trying to pour a rather large volume into the stomach. Because if you're exercising and slopping around, it can be quite difficult to do your best when you've got a full stomach, depending on exercise. Cycle exercise is obviously easier than running exercise. Cycling or skating exercise seems to be, that gliding effect seems to be less of a problem with gastric fullness. So, I mean, I think each athlete has got to work out the best rehydration strategy they've got during exercise, and probably pre and post as well. But I mean, I don't think as many people can exercise really hard on a full stomach. There obviously are some, but I think it's limited in that.
Could I ask you first of all about the age of the people in which these studies have been done? My guess is it's probably predominantly lab volunteers. And the context of my question is a clinical one, which is that we spend a lot of time looking after older people who are recovering from severe illness. It may not be a GI illness, but they're nutritionally challenged, and we spend a lot of time giving them protein, energy, liquid supplements to try and reinforce their nutrition. So my question is whether there are any lessons we might learn about how we should best administer that in order to get them to absorb it, because it's a crucial part of our management strategy to try and help these people recover.
Yeah, good question. Certainly, majority of the studies that have looked at gastric emptying and intestinal absorption, relatively young people. We've used some subjects who are in their 30s, but the majority would probably have been, 18 to 25 type of age. I can't think of any studies off the top of my head that have looked at people, 50 plus. I think using isotopic tracer would be an ideal method for doing it, because there are only certain people who you can intubate comfortably, especially with the perfusion tube. But an isotopic method, I think, could be used in older people. And being one of the older people, maybe I should volunteer. But I'm not aware of any studies that have specifically looked at older people. We know the thirst response is ablated slightly, and we know the kidneys don't function quite as well. I know that as a personal fact. And I really can't think of any study that's looked at that there. And therefore, it would be quite difficult to sort of be positive about methods one could use to look at people, especially people who are ill, to drink. I mean, I can remember when I worked in a unit that was part of the professorial surgical unit there, the encouragement that the nursing staff tried to get people to drink, but it was mainly water and tea. That was the big thing to get into them. It wasn't, if anything else other than that, it went in IV on it there.
But you remember too, John, in that surgical unit, we suggested that people might be able to tolerate oral fluids after surgery, whereas the surgeons all said, once you've had a general anesthetic, forget it, it has to be IV fluid, and I'm sure Ahmed will have some experience there. We found that people could tolerate oral fluids perfectly well, so the gut was very functional, even very soon after major surgery.
Crystallize my question a bit more, and I think I may know the answer, which is that we don't know. But so you've got an older person, they're ill, their gut works. In front of them is their trade name 40 juice or whatever it might be. Do we tell them to glug it very slowly over 4 hours, a sip at a time? Are they better to try and get it down quickly? Do we have any information about what will maximize absorption of nutrient or fluid in that setting?
Well, what I would say is if you take the effect of cholera, what they've in the developing countries there, what they have used is a sip technique throughout the day, where either the patient or a helper has teaspoon by teaspoon fed the same, and that seems to be very, very effective, given that you've got the time to take it in. And in today's NHS, you can still bang in an IV just to make sure people are well hydrated, and then I would suggest that it would be a steady sip. The difficulty being that if the patient can't sip it, you're not going to get nurses spending the time making sure that they will get that fluid in. So in that instance, then it might have to be glug it down quickly, and you're more likely to get people vomiting in that situation there. I think in illness, and as a general thing, I think in illness and in trauma, it's the stomach that is the weak point. That's the bit that seems to get most upset. As some of the studies that I've put in my review paper, they showed that people who had been injured in the Korean War, if you got a tube into the small intestine and put fluids into there, they were absorbed reasonably well. It was the stomach that seemed to be the major problem there, the weak point.
We'll come to Ibrahim and then to Phil. I think you had your hand up after that.
Well, sorry, two short questions. So the one is the influence of alcohol, uptake of water from alcoholic drinks. Is it facilitated? Is it less?
Depending on the alcohol content, but certainly if you have dilute alcohol drinks, they seem to be absorbed quickly. I don't think there is the same osmotic effect produced by the alcohol being absorbed as there is from carbohydrates or proteins. But you seem to get fairly, I mean, it's at least as good as water, it would appear. And people can hydrate using dilute alcohol drinks. And they very happily seem to be able to hydrate in that situation there. Perhaps if they have some crisps along with it, so they've got a little bit of carbohydrate and protein and a little bit of salt, that maybe help it as well.
Independent of the temperature of the drink?
I mean, I don't think for most beverages temperature is a big effect, has a big effect on gastric emptying or intestinal absorption. If you had something with a fairly high thermal capacity and you were drinking far large volumes of it, then that's going to have an effect. As Ron was saying, that they had some problems with athletes who were being asked to drink 1 to 2 litres of a very cold drink. Then they found it quite difficult to drink that sort of volumes that cold. I don't have any experience with that sort of volumes, but for volumes of around about 600 mils, it doesn't seem to be that difficult to do for individuals.
I tend to link the absorption or uptake of fluids to the requirements, water requirement, fluid requirement.
I wouldn't say that's
no.
No. I mean, most of us drink more than we need to drink because we enjoy the taste, or we're into that habit of when we drink, you urinate out the excess as long as your kidneys and bladder are in good condition.
Okay, yeah, but this leads to next question. Is there an optimal composition of the diet with regards to the water requirements?
Not that I'm aware of, not as far as requirement is concerned. I mean, I think it's dependent on the water losses, or at least the anticipated water losses, if you want to maintain reasonably good hydration status. But I think probably everyone here has got a slightly different habit of when they take in fluids, when they eat, how much they take in. Lots of people take.
In the general recommendations, so for water, so for healthy people, we say around 1 ml per 1 calorie, kilocalorie.
Yeah, I mean, I think that's an arbitrary figure that has got no. I mean, it's come from epidemiological studies suggesting that that's a reasonable amount, and it probably is what works out for majority of people on a daily basis.
But the energy we ingest is not only from carbohydrates, so we have a mixed diet. And
yes, and amino acids are equally as effective as a lot of the carbohydrates. What I'm not particularly clear about is the effect of fats in that they are absorbed in a slightly different way, and probably are not majorly responsible for producing those osmotic gradients that will allow water absorption. But equally, electrolytes are poured into the small intestine, and the uptake of those electrolytes the reuptake of these electrolytes can help water absorption. So fats, I'm not so sure about. Proteins and carbohydrates have a positive response to water absorption.
And salt?
I mean, depending on the amount of salt lost, I mean, you must, in order to maintain hydration, you must keep up reasonable salt levels in the body. But for the actual beverage or for the food, it doesn't necessarily need to have the salt there in order for water uptake. But on a day-to-day basis, the salt has to be there, otherwise it's not going to be taken up, and there's going to be major health problems if we get a drop in even circulating sodium levels.
Maybe just a comment on your previous point there, Ibrahim. If you go from an environment like this to a hot environment, you can increase your body water losses by 50%, but your energy requirement doesn't change. If we take an athlete to a hot climate, they can perhaps have 50% higher energy requirement than yours, but the sweat loss may be 5 liters or 6 liters a day. So you can completely dissociate the water requirement from the energy requirement. It just so happens that for sedentary people in a temperate environment, it roughly works out you can rub along okay on one mil per calorie. But it doesn't work for active individuals, and it doesn't work in hot climates.
Yeah, but the energy requirements do not vary as much.
Exactly. The energy requirement doesn't vary, but the water requirement varies hugely. So if that's true in a cool environment, it can't also be true in a hot environment. So, it just happens to work in temperate climates, which is where most of us live. We had a question from Phil next, I think, and then Peter, and then Ian.
I guess this leads on from Ron's comment there. Carl Gazolfi's group showed that fairly hard exercise in hot environments can induce an increase in intestinal permeability. Is anything known about how that affects the delivery of water and carbohydrate or any macronutrient into the circulation?
I mean, it didn't seem to affect anything in Carl's group. In their perfusion studies, the perfusion rates were as you would expect from that group there. So exercise in a hot environment didn't appear to do that there. But I mean, it is known that lipopolysaccharide can be found in the circulation of individuals who've run very hard marathons, which suggested that there was increased permeability, and some materials such as E. coli were perhaps getting across the mechanical barrier of the small intestine. So I think permeability under stress, whether it be exercise or exercise in heat stress, will alter permeability. But that I couldn't see that slowing down water absorption, but it may in fact increase the net absorption, given that the tight junctions in between the columnar cells do tend to become more permeable when there is nutrients there and that are actively absorbed. And if overall permeability is increasing as well, then that would suggest there would be an increase in permeability, uptake. Yeah.
We have Peter, and then Ian, and then Richard.
Thank you. May I go shortly back to the question of 1 milliliter for 1 kilocalories? From a pediatric point of view, we think always for constant body weight, constant circumstances, but in the children, growing children, it was described in 1957 by Halliday and Seagal, and this is based on physiological experiences that with growing energy expenditure, the water loss is increasing. So for an infant with 100 kilocalories per kilogram body weight energy expenditure, we calculate 100 milliliter per kilogram body weight. So it's much higher than for us.
Right.
So I think this rule is still true for the maintenance fluid. Of course, the high temperature, extreme circumstances are more important factors to influence the needs for fluid. But this one-to-one rule is true. We should accept it, I think. And if I could have a question back to the glucose sodium co-transporter. So why is it discussed that the fluid should have some sodium? This is because the higher osmolarity of the fluid if we add sodium, or what's the reason for this? Because again, as a pediatrician, for me it's clear that sodium in the fluids is necessary. Yeah.
Yeah. The glucose sodium co-transporter increases the osmolality on the basolateral area in the gut, and it's the osmotic effect. So you've got there's usually one molecule of glucose being absorbed with 2, maybe 3 sodiums. So there's a rapid increase in the osmolality of the tissue in the basolateral margin during absorption, and that seems to be the effect that drives water absorption, the osmotic effect there. It goes from something round about, normally about 400 milliosmoles, studies that have looked at, up to 600 to 1,000 milliosmoles if you've got glucose transport along with the sodium. So it's markedly hypertonic during absorption.
Ian, you're next, and then we have Ian, Richard, and then David.
This sodium glucose co-transport is fine when you've got fluids, drinks alone, rather than drinks and food. But as soon as you start to have foods with sodium in the foods, and the presence of food will stimulate a lot more intestinal secretion than your drinks will. Mm-hmm. So then you get a lot of sodium in the intestinal secretion. So how much do you think it's important to still have sodium in the drinks?
Well, I think I said in my talk, I don't think it's particularly important for absorption, but it might be important for maintenance of hydration levels if an individual is losing sodium. I mean, they'll be losing it in the feces, yeah. They'll be losing it in urine. Yeah. So it's got to be replaced. So, but it doesn't necessarily need to be there for uptake.
And has anybody done the calculations to see what the consequence of the population adhering to the sodium recommendations might be?
Not that I'm aware of. I really think,
you know, because if we did, well, the American target is even lower than ours. Yes. But if we do get down to the sort of levels that are advised as maxima, not as averages, then the impact could be quite substantial.
I would agree.
So the real question I wanted to ask was this is all about hydration or maintaining hydration of people in exercising conditions, and certainly in the morning after an overnight fast when there's no, you've got these rapid rates of water uptake. But in a 24-hour period, most of the water uptake is not going to be that rapid, accelerated slope there, because you're going to have food and water in the whole of the intestinal tract, and the absorption is going to be much slower. If you take a water turnover of 2 liters a day, say, what proportion of that water turnover is described by this rapid acceleration here, and how much of it actually is just happening in the other 23 hours of the day?
Well, undoubtedly, for most people, it's a long, slow process of absorption. But if you drink anything, you will get a fast level of emptying immediately, no matter if you're taking it with a biscuit, taking it with food. Yeah. There is an element of some of that liquid will quickly move out of the small intestine into the small intestine, and you will get that effect there if there is absorbable nutrients present. And if you are eating, there would certainly most likely be carbohydrate and protein. So yes, I think that does occur. But how important is that? Probably not very important. But it is important if people are looking to rehydrate rapidly using beverages. Yeah.
Just a comment on your point about salt intake. We've measured the salt losses and sweat of athletes in various situations, and especially in professional football players. And we see some players who can lose 10 grams of salt in a single training session, training twice a day. So that's 20 grams of salt. Now, when we have gone on record as saying these people need to eat a lot of salt to maintain hydration status, to maintain fluid balance, I get abusive letters from your friend Graham McGregor, who tells me that the only reason they excrete a lot of salt in the sweat is because they eat too much salt. Now, my answer to that, of course, is that if it is the case that the salt gland is dumping salt, the sweat gland is dumping salt, which it can do to some extent, then they should also have an enormously high sodium content in the urine, because the kidney is going to be the primary vehicle. These people tend to have fairly low urinary sodium outputs in a day. So the reality is intake is struggling to keep up with loss in that situation. And if they did follow the population guidelines on sodium, they would have difficulty if not find it impossible to do the exercise they do on a daily basis. So I think there are some situations where an exception to the SALT guidelines are absolutely required.
We need to do the calculations for the general public. When you come down from 10 grams a day, which is, well, actually the UK male average is now 9, female is 8, but it was 10. And we get down to 6. You know.
Yes.
That will be interesting in terms of hydration status.
Indeed. Yes. Yes.
Richard, I think you have the microphone next.
Thank you. Well, you already mentioned that there are almost no data on the elderly that and their gastric emptying and reabsorption. So this would be very interesting because we have elderly people taking a lot of medications, and most of them give some dryness in the mouth, which stimulates water intake. Unfortunately, most of these people shouldn't drink that much because of cardiac problems, renal problems. Can we train the digestive tract to slow down water in air absorption? Because everything we talk, and especially in pediatricians, in pediatric units, we try to increase the reabsorption. Can we slow down the reabsorption or stop it even, both on the apical or on the basolateral receptors, whatever? Are you aware of something like this, or can we train behaviorally the digestive tract not to absorb that big amount of water?
It's a very good point. I mean, the majority of studies that have looked at training the intestine is to increase absorption. But presumably you can down-regulate these carriers. If you can up-regulate them, you should be able to down-regulate them. But how you go about doing that, I'm not quite sure. On that.
Curiosity, 1% of my patients, 1% of people cannot drink water because their kidneys don't function. They're on dialysis. So
yes.
It's important for them. And they are thirsty, trust me.
That's right, yes.
We have David next and then Jane.
Could we come back to the individual differences? You've emphasized that there are patterns of what we drink, when we drink, and how much we drink. But how much is it due to individual differences in physiology?
Well, is it physiology or is it their eating habits? We don't know. I mean, but what we can say is that if you do measure an individual on a day-to-day basis, and we've tended to have quite strict guidelines, and most people do, for these sort of studies, in that people can't have eaten, it's they're overnight fasted. They've got to eat the same meals in the days leading up to the study as they did the last time. So we're trying to prevent as many disruptions to the study that we possibly can. So to a certain extent it is an artificial subject that we're using. Because most people do eat the same things at the same time most days, but we do try and control for all of these factors. So the actual day-to-day variability in individuals out in the population, well, I don't think we've got any idea of.
You've said several times that you think the pattern of eating might entrain the body to react in a certain sort of way. But the only studies you quoted suggest that wasn't true. Is there evidence that it is true?
Yeah, I mean, we know that you can upregulate carbohydrate absorption by giving people large amounts of carbohydrate. We know you can do the same thing for fats and amino acids. I mean, the gut is trainable to a certain extent. So, but people's eating habits tend to be fairly similar on a day-to-day basis, unless their environment changes and they come to somewhere like here to eat.
Have people looked at polymorphisms or any other genetic?
Not that I'm aware of, and I haven't even seen any twin studies that have looked at these sort of things. I must admit. Sorry.
Thanks. I wanted to ask you, what are the implications of what you've told us for beverage choice in a healthy person? Does it matter what you choose to drink if you want to stay well hydrated?
My answer to that would be it depends what you're doing and what your lifestyle is. Obviously, if somebody's exercising and is looking to exercise again fairly shortly, then they're going to want to get rehydrated as quickly as possible. But for the average person who's living their normal life, this probably doesn't really influence them at all. And beverage taste is probably the main factor that determines how much a person drinks on a day-to-day basis. But we do tend to be creatures of habit, where we drink a cup of coffee in the morning and so on, and we eat and drink the same things at meal times, and we have episodic intakes that are pretty regular. May change at the weekend or if we go on holiday, this sort of thing. But most of us have established our own intake over long periods. And we know that with children, changing flavours, changing the look of the drink will encourage them to drink more. And so presumably you can make the drink taste quite horrible and there'll be less of it drunk. You could do that sort of things. But I think for the majority of people, they survive and survive quite well. I'll be interested to listen to the fluid intakes, the talk on the fluid intakes throughout the European community to see how these differ and in what way these differ. Because obviously different communities have different habits, but we seem to manage to stay fairly well hydrated most of the time and get enough energy in most of the time. Thank you.
Everybody's happy. So what are the key points you would emphasise then, John? Maybe. Oh, Phil. Yeah.
I don't know if this was asked when I had to nip out for a comfort break, but Asker Eugendrup's group has done a variety of studies looking at ingesting very large amounts of carbohydrate during exercise. Ingestion rates up to 2.4 grams of carbohydrate per minute. And they're looking to maximise oxidation of that carbohydrate. They don't often mention gastric emptying or feelings of gastric fullness. Yeah. All those studies tend to be cycle-based studies. Is that a big flaw of that research? And they make a lot of recommendations suggesting carbohydrate intakes of 60 or 90 grams an hour.
Yeah, yes.
Surely that's coming at the expense of fluid absorption to some extent.
Well, I don't know if it is. I mean, it would the rate of fluid absorption, yes, certainly. But if you look at their studies, I mean, they're looking at oxidation rates. They're not looking at, in a lot of the studies, not performance times. There are some with performance times, but they're not looking at performance as such. They don't look at hydration status in these individuals who are exercising quite hard. I mean, they're specifically looking at uptake of carbohydrate and the oxidation of carbohydrates. And they seem to give fairly large fructose contents to the subjects. And I know certain people in this room here who've had bad effects of eating too much fructose or ingesting too much fructose while exercising. So it's difficult to say with Asker's stuff the hydration status of the subjects, but given that they're drinking quite a lot and that they are exercising quite hard, I suspect that they don't urinate very much when they're finished. But if you looked at their hydration status, I suspect the total body water wouldn't be greatly affected by even the amounts that they're given.
And of course, all of those studies are cycling studies. If you're putting
that in when people running, it would be probably a lot more difficult for individuals to cope with that. Yes, yeah.
Thank you, John. I just made a couple of notes of things that, for me, were some of the take-homes. Right. And one of them was the fact that so many of the studies are carried out in a slightly artificial situation, i.e. overnight fasted and without the presence of food in the gut. And of course, we have to interpret things in the context of a normal lifestyle when food is consumed at regular intervals during the day. Most of the studies are done on healthy, young, normal males rather than females. Yes. And of course, there may be implications of menstrual status, and because we know that sex steroids influence various aspects of fluid balance. But we also need to look at older individuals, people with renal insufficiency, people with hypertension on medication, other contexts too. We need to try to understand the individual variability in GI function. Yes. Is it genetic? Is it lifestyle? Is it something that happened in the early development? And we need to think about how all of this impacts on, terrible phrase, that impacts on, I vowed I would never say that, how it affects beverage choice and how individuals balance the hydration with the other things that come from drinks, the energy, the vitamins and minerals you get if you consume milk or whatever. Yeah. So those for me were some of the key messages. Would you?
And I think there's also another one there is that we need to look at children as well.
I'd include those in the other populations, yeah.
Mm-hmm. But I would also say that the isotopic method lends itself to being used in bigger populations and mixed populations. And I have looked at uptake of deuterium into the blood, but you could also obviously look at it in the breath. Yeah.
If it's used properly and interpreted correctly, because there are people in the literature who've dismissed the technique because they haven't understood what it's telling you.
Yes. Yeah, indeed. Yes.
We have a challenge. I mean, the ideal is to measure it throughout the day. And the problem is that if you have deuterium in each dose, then you have an accumulating background against which you're looking at the spikes.
Yeah, but you can then subtract the background from it.
But the other thing is that you get condensation, metabolic reactions going on.
Yes.
So it stops being a marker for total body water.
But if you use mass spectrometry, you can have low levels of isotope being used.
But you can also switch between deuterium, tritium, and oxygen 18, so you can swap your tracers around.
Yeah. I think one of the other things that probably should be looked at as well is you perhaps should be labeling the total body water with one isotope and giving another one to let's see what the various fluxes occur.
Well, we did some of those studies, but never published it. Yeah. So you can go home and write up some of those studies.
Yeah. Thank you.
Because one of the things that was absolutely striking, particularly during exercise, if you give somebody a drink containing deuterium oxide during exercise and you've labeled the body water pool with tritium, if you inject tritiated water into a vein, the disappearance rate of that tritiated water during exercise is astonishing. It's down to equilibration within simply less than 2 minutes, and in some cases within about 30 seconds. Whereas at rest it takes you 30 or 40 minutes to equilibrate. During exercise, bang, it's very, very fast. So you can do quite a lot with using more than one isotopic tracer.
I'd have to say that the modelling of it is an important factor as well, and getting somebody who is really reasonably efficient and effective at modelling these things would be paramount.